So what we're going to look at now is for an individual who has this heterozygous genotype, how is it that the different alleles assort independently when we look at then the chromosomal aspect of it through meiosis, right? So as gametes form and then the alleles go into the gametes, how does that pattern actually work to give us all those options, right? Because remember, we're going to have options where we need to have options in the end here, where we have gametes that can be carry both the dominant and a recessive, and then the other way around. Like this. Okay, so we need to see all these options and how they will turn out. And so this is what we're going to do here. So the pink one here, the reddish one, represents one particular chromosome. The blue one represents a different chromosome. This chromosome here, the pink one, is the one that has the color gene on it. And then this one here is the one that has the height gene on it. Okay. So DNA replication will occur, and then you're going to get two of those and two of those. Same thing here, two of these and two of, I'm just going to label them just so you know which, not exactly where on the chromosome they are, but just to know which one it is that we're following. Okay. Now, what will happen is then they'll separate into different cells. So there'll be cell division, the first division of meiosis in meiosis 1, and then we'll get pulled onto this side. Let's say the R, this one here, kind of goes this way, and then the TT, the way I drew it out. So the two dominants went over here, and then the two recessives went over here. You can see that. So these are representing the recessives. Now there'll be another division. So this cell will divide, this cell will divide, and then you'll get, that'll split, and so you'll get single chromosomes of each type, and then you'll get the R, the dominant. Here, this will have the R, the recessive. This will be dominant. This one will be recessive. So what we see here is only really the two possibilities. We only see the possibility for the dominant being together and the recessives being together. So here and there. What we need to see is how do they then pair up with dominant and recessive and the other way around. So what we're going to do now is look at the slightly different. Same basic scenario, but a different sort of arrangement. Okay, let's say this chromosome here is the one with the dominant, just like we had, and this one here is the one with the recessive. And this one here has the recessive, and this one is the dominant. Okay. Now what's going to happen is, so this one doubles up. You get the two of the dominant here, and then the recessive like this, RR, and then on this side, though, we're going to put recessive, dominant. So if you could tell, hopefully you can tell what I did different here. Okay, so this half now has basically, you have homologous chromosomes pairing. That's the same as over here. So the ones that have flower color, the ones that have height. Oops, that's what I did wrong. I knew I was doing something that looked very strange to me. Okay, yeah, this is supposed to be that. So this, keep doing it. There we go, like that. So same thing over here. Homologous pairing up, the ones that have flower color, the ones that are displaying height, the ones that have flower color, the ones that are displaying height. How I'm arranging them different here is that on this side over here, in this particular example, I have both the dominants, okay, on this side, and both the recessives on the other. But how they line up, they're separate chromosomes. They're different chromosomes. They could line up any particular way. So in this example over here, I have the dominant on this side for the flower color, and on the same side is the recessive for height. So here's the capital R for the dominant for red, and the lowercase t for the height for the short, right? As the way that they're going to get pulled together, they could happen one way or the other. So this could happen, or this could happen at an equal frequency. That's what we're talking about independent assortment. The two different chromosomes, they're independent from one another. And we're going to talk about also genes on a chromosome next, but we're just kind of simplifying it this way first. So what would happen then is we get the first division of meiosis 1, and then this particular cell here has the chromosome with a color, flower color gene, and it's red. And then also the chromosome with the height gene, which is short. And then over here we have the flower color gene, which is white. And then the height gene, which is tall. Now another division will take place. The second division in meiosis will occur. And then they'll line up and then they'll split. And then you'll get genes for flower color, genes for height. But the way they'll be distributed will be the dominant for red with the recessive for short. Red, short. And then you get white, tall, white, tall. This is tall. Right, so now here we have red, short. That's this one. And here we have white, tall. That's this one. So we see all four, right, of these possible combinations where the dominants are together, the recessives are together, or a dominant and a recessive pair up in different combinations. And that way, when we then do the test cross and say that when we do that Punnett square, I'll just do a super quick little thing here. When we do that Punnett square, and we assign these four possibilities, we said that one possibility is having the red and the tall. One possibility is having the red and the short. That's over here. One possibility is having the white and the tall. That's over here. And another possibility is having the white one and the short. It's right here. Equal possibilities of any of those being given into a gamete that would then go on to fertilize the offspring. So you can see it's not that we see more of the reds and talls in that 9-3 to 3-1 because they just exist more. It's because one is dominant, so it's masking all these recessive ones. But the way they're being passed on to the offspring, the parent has a 50-50 chance of giving the dominant allele or the recessive allele. And then the gametes that would be formed are going to be formed in equal proportions to one another. The reason we see them in different proportions is because the dominant's recessive, right? Not because of that they or more frequent, or less. That'll be a separate thing that we're going to talk about eventually is allele frequency in populations, as we get to how this then ties into evolution and evolutionary change. But it's important to understand some of the genetic basis. A lot of people don't understand the genetics sort of behind that and just kind of jump into the concepts. And without this sort of supporting it, then people have a lot of questions. Say, well, how do you know that? How do you know this? How do you know that? So this is how. This is part of the picture of the story of how you will understand change and then selection and that sort of thing over time. So this is sort of the chromosomal basis for the independent assortment, and what we have. All right.